today we start with the next part with respect to mesosome formation. Mesosome formation, it is totally absent in case of eukaryotic system. So whatever mesosome part is there, we have to focus on the bacterial cell in case of the prokaryotic system. In case of the mesosome formation, okay. So in case of the mesosome formation, basically we are talking about mesosome, nothing but these are the something plasma membrane ingrowth or folding structure. So simply we are talking about mesosome. These are the plasma membrane ingrowth. PM for the plasma membrane, and now we are talking about the ingrowth. So in case of the bacterial system, what happen? Suppose these are the outermost boundary, and at the some places that plasma membrane it will be start showing folding. They are showing ingrowth. So such typical structure these are called as the mesosome. And I think in chapter biological classification under the kingdom Monera, we already discussed detail two types of the mesosome, central mesosome and the lateral mesosome. We are not taking that part. Simple meaning, mesosome it is present in bacterial cell and it is a part of the plasma membrane, but plasma membrane they are showing ingrowth, right? And due to their ingrowth, ultimately they can form a structure, the typical structure called as the mesosome. Number one, mesosome, they are help in DNA replication. They help in DNA replication process, first function with respect to mesosome. Because now what happened, you know very well, in case of the bacterial system, nucleus is absent, right? And inside the nucleus, genetic material is present, DNA present, but in case of the bacteria, such nucleus is absent. That's why, purposefully, in case of the bacterial cell, mesosome structure is present. Such mesosome, they provide attachment site for the genetic material, attachment site for the DNA, right? So you know very well, we just write down one sentence. In case of the bacteria, genetic material or that DNA, they attach to the plasma membrane because they float nahi hota. They never float inside the cytoplasm. They should attach to any site of the plasma membrane, and that site specifically called as the mesosome site. Mesosome site, they provide attachment site, they provide binding site. That's why genetic material or DNA they can easily hold at that mesosome area. And the function, first function with respect to mesosome, they help during the DNA replication process. Another one, further we discuss such kind of the some kind of the respiratory enzyme also. Respiratory enzymes, they are also present on mesosome because again mesosome they provide surface area, they provide attachment site, and if you want you can write down some example like the enzymes those are involved during the ETS system, that is electron transport system. Okay, because you know very well under the ETS, under the electron transport system, major organelle that is mitochondria, but in case of the bacteria mitochondria absent. So plasma membrane they provide attachment site for the enzyme, and we are enlisting few enzymes like the enzymes those are involved during the ETS system, that is electron transport system. Okay, yeah. So any doubt from the mesosome, any doubt from mesosome? Okay. Yeah. After the mesosome, we are talking about fimbriae and the pili. Next one, number fifth, pili and fimbriae. I hope you remember pili and the fimbriae. Again, these are the characteristic feature which is present only and only in case of the bacteria, while in case of the eukaryotic system, they are absent. Hmm? Now, when you talk about such condition, pili and the fimbriae, so first point, basically they are some kind of the outgrowth. They are outgrowth. Mesosome basically they are ingrowth, but when we are talking about the pili and the fimbriae, they are outgrowth. And when we are talking about what is the composition, so simply these are made up of protein. Name of the protein that is pillin protein. So both pili and the fimbriae They made up by the protein, and name of the protein, that is pillin protein. Okay, pillin protein. Now what happened? In this condition, we are talking about the common function. Flagella basically they responsible for the locomotion, movement. But when we are talking about the pili and the fimbriae, they are having some hair-like outgrowth or some bristle-like outgrowth. So major function with respect to pili and the fimbriae, they help in attachment. Question. They help in attachment. Okay. Flagella also present, but flagella major function locomotion. Pili and fimbriae they don't involve during the locomotion. They having only one function. They help for the attachment process. Got it? Question already asked. Chalo. After the pili and the fimbriae, next one continue with the flagella. Yes, in case of the prokaryotic system, flagella present, as well as in case of eukaryotic system, flagella is present. But we are talking about exactly difference. In case of the pro, flagella made up of protein, that is flagellin protein. They made up by the protein, which protein? That is flagellin protein. And when you compare this point with eukaryotic flagella, so eukaryotic flagella basically they are made up by the tubulin protein. Okay, and later at the end, same in this chapter we'll discuss eukaryotic flagella. They are showing 9+2 arrangement. They are also showing 9+2 arrangement. So what is meaning of the 9+2? Entire portion we'll discuss in this chapter, means during the upcoming classes. 9+2 means means it is made up of the tubulin protein. So 2 tubulin protein they present at the center, and the 9 they present toward the periphery. 9+2 arrangement. So we'll discuss that part later. And again, one question coming from such kind of this arrangement. Okay, Abhilasha. Just check. We just started from mesosome. One student, please tell me, okay? एक minute, Pratyusha. Which part is flagella present in eukaryotic cell? See, when we are talking about the flagella, flagella is consist of the three parts. Basal, that is basal plate, again, and the filament. Basal plate, these are the common part which is present in prokaryotic flagella as well as the eukaryotic flagella. Okay. Suhas, cell wall, we already covered that part. Cell wall part we already covered. Got it? थोड़ा थोड़ा हम cover कर चुके, detail भी discuss. Yeah, once we finish this part, then we discuss more detail. Okay, Abhilash. ठीक है. प्रत्युषा, एक important question यहाँ से बनता है, that is related to plasma membrane. Means prokaryotic flagella, it has the plasma membrane or plasma membrane is absent. So that part we'll discuss later. Okay, chalo. Yeah. So we talk about the next part, number 7, with respect to difference between pro and the U. Cytoskeleton. You know very well cytoskeleton nothing but these are the such kind of the some protein plus fiber combination who provide stability. So cytoskeleton they are totally absent in case of the prokaryotic system, but when we are talking about such condition in case of this eukaryotic system, yes, cytoskeleton is present, and three types of the cytoskeletons are there. Number 1, microfilament. We'll discuss this part later in detail. Microfilament. Number 2, intermediate filament. Intermediate filament like keratin protein who provide this stability, extra stability. And the last one, microtubules. Okay, so three types: microfilament, intermediate filament, and the microtubules. And if I'm talking about the major function, means whatever we are talking about, the microfilament or intermediate filament or the microtubule, all those are collectively, it has the different function, but we are talking about one common function which is associated with this. They are responsible for, they provide support to the cell. They provide support to cell. Common function. Actually, it has the different function. We'll discuss that part later. Okay. And please remember, whenever we are talking about the cytoskeleton, cytoskeleton तो obviously present है, and that cytoskeleton, they are made up of protein plus fiber. We'll discuss that part later. Actin protein, myosin protein, or tubulin protein, etc., etc. So they are made up by the two components. Number one, protein, and the another one, fiber. Protein and fiber. So just quickly check. We are talking about the part of the cytoskeleton, which is totally absent in case of the pro, while in case of the U, it's present. Three types of the cytoskeleton who provide stability, who provide the support, who maintain shape, etc., etc. बहुत सारे functions we'll discuss the later. चलो. After the cytoskeleton, next one we are fiber formation in the cell division. Chalo, should I continue? Next part we are talking about with respect to spindle fiber formation. Spindle fiber formation during cell division. During cell division, you know very well. Spindle fiber formation, these are the characteristic feature which is present in case of the eukaryotic system. So yes, in case of the pro, it is totally absent, but in case of the U, it is present. Okay, so absent and the present. Next part, we are talking about sexual reproduction. Sexual reproduction. You know very well, for the sexual reproduction, two parents are required. One is male and another should be female. So such kind of this condition, it is totally absent in case of the bacteria, in case of the pro, while in case of the eukaryotic system, sexual mode of reproduction is present because male, female, they are present. But you know very well, when we are talking about, in case of the bacteria, sexual reproduction is absent, but parasexuality is present. Can you tell me the types of the parasexuality? But parasexuality is present. हाँ प्रत्युषा, वही पूछ रहे हैं ना. So we are talking about, yes, only transfusion. Three types of the parasexuality. Number one, transformation. Transfer of genetic material from one bacteria to another bacteria. Transformation. Number second, conjugation. Transfer of genetic material with the help of the conjugation tube. One is donor, another one is recipient. And the next one, that is transduction. Transfer of genetic material with the help of vector, with the help of the viruses. Okay. So these are the three types of, yes, these are the three types of the parasexuality where genetic recombination can be occur, but it is not a true sexual reproduction. That's what is we are saying. These are the parasexuality, which is very much common in case of the bacteria. Chalo, done this part. Done. Okay. ये हम पहले देख चुके हैं किंगडम मोनेरा. Yeah. So next point we are talking about number 10 point, presence of the true vacuole. True vacuole. Okay. True vacuole. So you know very well, true vacuole, it is absent in case of the prokaryotic system, but in case of the U, true vacuole is present. But we are talking about one presence of the gas vacuole in case of the bacteria. Which vacuole is present? In case of the bacteria, gas vacuole is present, and such gas vacuole, basically, please remember, it is not a true vacuole, it is example of pseudo vacuole. That is false vacuole. It is not a true. True means what? Those are responsible for the osmoregulation, excretion, etc. etc. So such type of the vacuole, these are the present in eukaryotic system. But in case of the bacteria, gas vacuole is present, but that gas vacuole is not a true vacuole. These are the vacuole, but it's a pseudo vacuole, false vacuole, right? And you know very well the function of that gas vacuole, basically they help in floating, they maintain the buoyancy, they help in floating, which is present mostly in aquatic bacteria, right? Got it? Okay. So please remember, true vacuole is absent, but in case of bacteria, gas vacuole is present, and that such gas vacuole is not a true vacuole. These are the pseudo vacuole, because obviously these are they such kind of the vacuole is present, but function, they help in floating, they maintain the buoyancy. Okay. Yeah. Next part, we are talking about another concept with respect to vacuole, that is cyclosis. Could you please tell me what is meaning of the cyclosis? Anyone? So we are taking the next condition. What is meaning of the cyclosis? So first हमने देखा true vacuole is absent, and the cyclosis eating really Vedant. Cyclosis process, it is absent in case of the bacteria, while cyclosis it is present in case of the eukaryotic system. So quickly tell me what is meaning of cyclosis. One question for me. Cyclosis, tell me what is the meaning of the cyclosis? Eating, cyclosis, flow of cytoplasmic smoothly removing. अरे ना रे बाबा. Flow partial. Deepika. Okay, थोड़ा थोड़ा correct है. Cyclosis means what? Movement of cytoplasm. Yes, it's a cytoplasmic streaming. Yes, movement of cytoplasm around the vacuole. So what is meaning of the cyclosis process? Cyclosis process nothing but these are the movement of cytoplasm. Movement of cytoplasm around vacuole. Around vacuole, right? And such type of this movement, that is also called as cytoplasmic streaming. These are also called as the cytoplasmic streaming, cyclosis. Hmm? So quickly we are taking the diagrammatic representation, how it is exactly look. Suppose ये कोई cell है. These are the one plant cell. Inside the plant cell, you know very well, maximum area it is occupied by the vacuole. So suppose these are the vacuole. Okay. Now what happened? Whatever the cytoplasmic material is present, they all cytoplasmic material, they are present toward the periphery in case of the plant. Even nucleus also, they are located towards the periphery, because maximum volume it is occupied by the vacuole, because vacuole, these are the largest organelle in plant. Okay. So now what happened? Whatever the liquid part, what is the fluid part, cytoplasmic part, which is present inside the cell, they are showing movement. So movement किसी भी तरह हो सकता है. Movement may be clockwise or movement may be anti-clockwise. Doesn't matter. किसी भी direction से movement हो सकता है. Okay. So such kind of the movement, that is cytoplasm, movement of the cytoplasm around the vacuole. Such type of the condition, simply it is called as the cyclosis, and cyclosis also called as the cytoplasmic streaming, which is totally absent in pro, but it is present in case of U. Got it? Yeah. So any doubt from the cyclosis? Plasmodesmata, it's a cytoplasmic connection. Pratyusha, have patience. We'll discuss that part quickly. These are the cytoplasmic connection. Cyclosis, it is a movement. Chalo. Okay, chalo. So next part we are talking about कितना number है ये? Number 11, genetic material. So next part we are talking about genetic material which is present in pro and the U. So in case of the bacteria, we are enlisting the name. Whatever we are talking about the genetic material, yes, in case of the prokaryotic system is present as well as in case of the eukaryotic system is present, but it has some various name. So whatever the genetic material present in bacteria, simply it has the different name. First of all, we are talking about name. Genetic material which is attached to mesosome, that entire genetic material collectively these are called as the nucleoid. Nucleoid. Okay? Or sometimes these are also called as the naked. So I'm giving the or symbol, so we'll get clear cut idea. So nucleoid also called as the naked genetic material. Why naked? Because nucleus is absent. There is no any protection wall. That's why it is called as the naked. Naked genetic material also called as genophore. Genophore, that is nucleoid, these are also called as the incipient nucleus. Incipient means what? Not a completely nucleus. जैसे हम कहते हैं ना कि nucleus it has the wall, plasma membrane, etc., etc. So that's why it is called the incipient nucleus. And such kind of the genetic material, these are also called as the prochromosome. Pro, ठीक है? Prochromosome. So these are the different name for the genetic material which is present in case of the bacteria. It is called as the nucleoid. It is called as the naked genetic material. Is called as the genophore. Is called as the incipient nucleus. Also called as the prochromosome. Hmm? But in case of the eukaryotic system, it has the very simple name. Genetic material basically is placed in which part? They are situated that present inside nucleus, because that specific compartment it is present, that is nucleus is present, right? And whatever we are talking about the nucleus, हम आगे देखेंगे, it has the nuclear membrane. So whatever the genetic material is present, that all genetic material it is covered, it is not naked, it has the covered genetic material. Next part, after the genetic material part, now we are talking about next organelle which is present inside the nucleus, that is nucleolus. Nucleolus, so you know very well, nucleolus it is absent in case of bacteria while it is present in case of eukaryotic system, because it's a dark body which is reside inside the nucleus. Next part we are talking about nature of DNA, number 30. Nature of genetic material. So obviously, in case of the prokaryotic system as well as in case of the eukaryotic system, genetic material it is present. That is, these are the DNA. It's not RNA. But in case of both prokaryotic system as well as in case of eukaryotic system, DNA basically is present double-stranded. Again, they are coiled. यहाँ तक common है, but variation with respect to their nature. So bacterial genetic material, basically these are the circular, while eukaryotic system, whatever the genetic material is present, that present in a linear form, right? Linear and the circular. And the last one, you know very well, DNA, it has the negative charge. So for the packaging process, we required positively charged amino acid, positively charged protein. So in case of eukaryotic system, there is a presence of the histone protein who has positive charge and which is responsible, which is helped during the DNA packaging. While when you compare this point with pro, in case of the pro, there is a presence of the polyamine rich protein. Polyamine rich protein, because histone protein is absent. But common characteristic, again it has the charge, that is positive charge, because again it has the negative charge. For the packaging process, for that purpose we required positively charged protein which is helped during the packaging process. Okay, so difference with respect to two parameter, number one circular, linear, number second what kind of the proteins are present. That's it. Chalo. Now next part we are talking about exactly location for the DNA. So you know very well, in case of the bacteria, whatever the genetic material is present, they are present in common cytoplasm. सब कुछ वही होगा because there is no any separate compartment. So in case of the prokaryotic system, Abhilasha. Okay. So in case of the prokaryotic system, whatever the DNA is present, that whole DNA it is present in cytoplasm in common compartment. Okay, it is present in cytoplasm. But when you compare this point with eukaryotic system, so in case of the eukaryotic system, it has the different location. So which kind of this location? Yeah. Okay, thank you, Abhilasha. So which kind of this location? In case of the eukaryotic system, first of all, genetic material present hoga in nucleus, first one, right? As well as we are talking about genetic material, that is DNA, also present in mitochondria. We'll discuss that part quickly. So again, these are also present in mitochondria. And again, genetic material, these are also present in chloroplast also. So in case of the eukaryotic system, these are the three organelles: nucleus, mitochondria, and the chloroplast where genetic material is present. Got it? Okay. And you know very well, whenever we are talking about the genetic material, obviously central dogma, that is replication, transcription, translation, all these processes are carried out in nucleus. Means हम आगे देखेंगे replication and the transcription in nucleus and the translation in cytoplasm. Similar for the mitochondria also and the chloroplast also. Okay? Yeah. So last point with respect to site. Site for, first of all, site for replication, then site for the transcription, and the last one, site for the translation. So replication means from the one copy of the DNA we form the two copies. Transcription means from DNA we can form messenger RNA, and translation means formation of the protein. So all these processes, they occur simultaneously in common cytoplasm. So they are present in cytoplasm. But when we compare this point with eukaryotic system, so replication as well as transcription, these two processes, they occur inside the nucleus, while the process of translation, they are occur inside cytoplasm. Done. Okay. Special condition we'll discuss later. Means in case of the mitochondria and the chloroplast also, inside those organelles, ribosome is present. So that is a exception case we'll discuss later. These are the some common point with respect to difference between the prokaryotic cell and eukaryotic cell. Yeah. Okay, chalo. So any doubt from pro and the U? Any doubt? Just quickly check. If you have any doubt, you can ask me. Okay, chalo. So remember one thing, whatever the questions are coming, that question you have to compare this point, because ये जो point है ना, this prokaryotic and eukaryotic cell, it's coming again and again, because next year also microbe chapter है आपको. During the microbe also you have to remember this. Even in case of the biotechnology, आपको Cinderella से means Cinderella means E. coli bacteria. So you know the what is the exactly E. coli bacteria, then the another bacteria which is responsible for the TI, tumor inducing factor. I think Agrobacterium tumefaciens, right? So that bacteria also get repeated. So whatever the part is there, common characteristic you have to remember, because it will be help for the bot as well as the zoo 11th as well as 12th standard. ठीक है. And again बहुत सारे sodium potassium pump होंगे, active pump, passive pump. That time also you have to remember in which organelle calcium is more, in which organelle means other minerals are more. So according to that. Okay, fine. So all these are the difference with respect to pro and the U. Now we are talking about one by one organelles, and now we start with the first organelle, that is cell wall. First part, cell wall. Discoverer of the cell wall, you know very well Robert Hooke, because Robert Hooke, the first scientist who observed dead cell under microscope. So whatever the what we say contribution of discovery of the cell wall, it is given to scientist Robert Hooke. Now we are talking about first condition in case of the plant as well as in case of the fungi, in case of the prokaryotic cell as well as in case of the some protista, typical cell wall. For the cell wall, I am using CW. Typical cell wall is present. But in case of the gametes, male gamete, female gamete, or in case of the zoospore that is motile spore. I hope you remember. As well as in case of the animal cell. In case of the animal cell, such type of the cell wall is absent. I think आपको animal cell ही पता होगा, but please remember during the gamete formation and during the zoospore formation, cell wall is absent. ठीक है. Now, first part with respect to cell wall, we are talking about composition. Composition of cell wall. When we are talking about the cell wall, inside the cell wall, major composition that is matrix. Another composition that is microfibril. And the third composition that is deposition. So each and every cell wall, they made up by these three components: matrix, microfibril, and the number third, deposition. Okay. Now when we are talking about the matrix part, matrix nothing but these are the something a gel-like substance. It's a ground substance, gel-like ground substance. Okay. And whenever we are talking about the matrix, this matrix they consist of. So I'm giving the first part. Matrix. Inside the matrix, basically, सबसे ज़्यादा concentration होगी water की. Water is present in high amount. Followed by there is a presence of the hemicellulose. Followed by there is a presence of the pectin also. Followed by we are talking about lipid concentration. And finally, there is a presence of the concentration of the protein. We are not taking concentration. If you read module, आपको बहुत सारा detail में मिलेगा, but no need. First of all, we are talking about part that is matrix part. And in case of this cell wall composition, matrix, these are the major component. First one is the major component. And out of that, water they are present in a maximum amount. ठीक है. Done. So first of all, we talk about that is first part that is matrix, which is a major component, which is rich with the water at high abundant amount, hemicellulose, pectin, and lipid followed by protein. Next part we are talking about second component that is microfibril. When we are talking about the part that is microfibril basically it's a second major component. And if I'm talking about the percentage, is having approximately 20 to 40%. But we are taking few important points. When we are talking about the plant microfibril, plant microfibril. So such plant microfibril simply they made up of material that is cellulose material. Plant microfibril. Okay, they made up by the which material? Cellulose material. Another one, when we talk about the fungi, so fungi microfibril, now they made up by the material that is chitin material. Okay, and whenever we are talking about that microfibril, so microfibril, it has the common function. Basically, they are structural elements of cell wall. So whatever we are talking about microfibril, they are having the important function. These are the structural elements. You know very well, structural elements means what? They maintain the shape, they maintain the size of the cell. So जैसे हम कहते हैं ना कि plant it is cell wall, it is rich with the cellulose. In case of the fungi, they rich with the chitin. So these are the part of the microfibril, and such kind of the microfibril they maintain, they act as a structural element of cell wall. That's it. Okay. Yeah. And the third part, we are talking about deposition. Deposition means what? It's not compulsory actually. In some cell, 0% deposition is present. मतलब कोई deposition ही नहीं होता. And some cell they might be present. So if I'm talking about the concentration, it is lie between 0 to 25%. Some cells they totally lack, and in some cell they are present. So if you want, you can remember this point. So it can be present or absent, dependent. Okay. So quickly we are taking the few examples. First part, first example, we are talking about lignin deposition. You know very well, lignin, these are the part of the deposition, and such kind of this lignin, they are very much common in case of this sclerenchyma, that is a dead tissue. It is present in sclerenchyma, as well as they are parts of the tracheids. Tracheids, nothing but which is responsible for the water conduction, as well as it is part of the vessel. And in case of the fiber like xylem fiber and the phloem fiber, such kind of the lignin material it is present. So whenever we are using the dead tissue, that time remember sclerenchyma, they are in case lignin they are get associated. It is present in sclerenchyma tissue, they present in tracheids, they present in vessels, as well as they present in fiber. And the major function सबका एक ही होगा, provide the mechanical support. And the tracheids and the vessels, these are the two types of the cell which is responsible for water conduction. So this part we'll discuss in another one. Next one, next material, that is suberin material. You know very well, suberin basically these are the present, mostly it is present in case of endodermis layer. I hope you remember, these are the present in endodermis layer of root, not in stem. ये बस कहाँ पे present होगा? It is present in case of the root endodermis layer. Yes, Pratyusha. And such kind of this layer, simply it is called as the Casparian band or Casparian strip. Simply these are called as the Casparian strips. Very simple. Okay, suberin material. And another example, such kind of this suberin material also present in cork cell. So one, endodermis of the root in the form of the Casparian strips, and the number second, these are also present with respect to cork cell. Third part, we are talking about cutin. Cutin basically, these are the one kind of the fatty acid which is form of cuticle. So basically cutin form a cuticle, that is a something of waxy layer. Cuticle means what? These are the something of waxy layer which is present on the leaf as well as the stem. Which is present on leaf as well as the stem. So reduce the transpiration, control the water loss on the leaf and as well as on the stem. Something of plastic layer, something of waxy layer is present. That layer it is called as the cuticle, and that cuticle basically it is due to some fatty acid substance deposition, that is cutin. Okay, and if I'm talking about the cutin function, they're responsible for reduce the water loss. That's it. Next one we are talking about, in some plant silica also get deposited. Silica. An example for the silica, obviously you know very well, grasses, and the another one that is equisetum. Grasses as well as the equisetum, right? And equisetum also called as, what is the common name for the equisetum, which is a part of Peridophyta? Equisetum also called as, equisetum also called as, yes, these are also called as the horsetail because it has very long structure, long elongated structure. Okay, so that's why it is called as the horsetail. Yes, true. So please remember, in case of the silica, whatever we are talking about, in case of the grasses as well as the equisetum, there is a presence inside the cell wall. Silica get deposited, and if I'm talking about what is the function of such silica, simply they provide stiffness. So that's why they involve during the protection. One second though. Write down function, provide stiffness. The screen is get lagging. Hmm. So silica deposition. So we are talking about function. Silica just provides stiffness because it's quite hard. That's why it is involved during the protection process. Okay. And the last point, number 5, there is a deposition of some material like calcium as well as the iron. And the best example that is Kara. Kara. This is a part of the green elf. हमने देखा था Kara, these are also called as the stonework because it is a deposition of the material like calcium and the iron. Exception case, who has the multi cellular as well as the jacketed sex organ, which is the exception case for the green algae, means entire algae actually. Specifically, we are talking about specifically in case of the green algae. So it is a part of the green algae that is Cara, where inside the cell wall deposition is present in the form of calcium as well as the iron. Okay, done this part. Done. This deposition part will help you in chapter anatomy. Because हमें सब कुछ repeat करना है. Lignin, suberin material, cutin one, even silica also. That will help you in anatomy chapter. Okay, chalo. Next, quickly we are talking about different layers of cell wall. Next title. Different layers of cell wall. So you know very well, whenever we are talking about this cell wall, it has the 3 layers. Number 1, that is middle lamella. Middle lamella also called as the cementing layer. These are also called as the cementing layer. Another one, primary cell wall, and the next one, that is secondary cell wall. Okay, primary and the secondary cell wall. So quickly we are taking one by one, simple diagrammatic representation. Suppose ये 2 cells हैं. One plant cell, and now these are the another plant cell. Sorry. Between the 2 plant cells, now there is a deposition of the some material. Surrounding that cell, now there is a deposition of the another material. And finally, we are talking about there is a deposition of the third material. So I'm just giving the color so we'll get the clear cut idea. So they are present all over the wall of the cell, giving the number one, number two, number three, 1, 2, and now these are the three. And whatever the green color is there, that green color part, that is a plasma membrane. So when you consider any cell from outer to inner, so number one layer, first one, that is middle lamella, which is present toward the outermost side, outermost. Now, inside the middle lamella, presence of the primary cell wall, and again toward the innermost side, there is a presence of the secondary cell wall. So in case of the plant, basically three layers are present, which is a three different layers of the cell wall. Number one, middle lamella, also called as the cementing layer, which is made up of the pectin. Inside the middle lamella, now there is a presence of the primary cell wall, and further there is a presence of the secondary cell wall, or finally आएगा हमारा plasma membrane. So first of all, we start with the number one, that is middle lamella. Middle lamella, basically, when we're talking about the middle lamella, they are also called as the cementing layer. Cementing layer, because now what happened? These are the, what we say, these are the common material which is present between the two cells. Cementing layer. And whenever we are talking about such middle lamella, so such middle lamella, commonly, they are made up of two substances. Pectin to common hoga, definitely. But along with that pectin, to increase the stability, now they associated with, they bind with the calcium pectate. First one, calcium pectate, or sometimes they get conjugated with magnesium pectate. Because when we are talking about the pectin or pectate, you know very well, these are the part of the some polysaccharide. It's a part of polysaccharide. Now to increase the stability, because the outermost layer है, आपने देखा होगा, for example, you see the tomatoes initially they are white in color, right? White in color. Slowly they convert into green, and finally they convert into red. So if you observe fresh tomatoes, they having kind of this some stiffness, they having the quite hardness. But slowly when that fruit, when that any tomatoes they are start ripening, ultimately they become a loose क्यों हो रहा है? Because from the outermost layer, which is a middle lamella, which is a cementing layer, that outermost layer it will be start degrading. So that's why if middle lamella or outermost cementing layer is strong, that time you can feel yes, fruit fresh hai or that vegetable is fresh. But slowly after a few days, that outermost layer, middle lamella, they start degrading. And once it is does start degrading, ultimately that fruit or that vegetable, it is become soft, right? So to increase that stiffiness, to increase that stability, pectate they associated, they bind with the calcium. So more condition, whenever we are talking about which is the more, so obviously calcium pectate they present at the more amount in cell wall, and magnesium pectate also present, but they are present in a less amount. So please remember, pectate they conjugated with calcium or magnesium to increase the stability. That's it, because it's the outermost layer, right? And once they start degrading, it means that now that ripening process it is get initiated. So ये जो part है, this part will discuss when we talk about chapter plant growth regulator. Okay, yeah. So these are the outermost layer, that is middle lamella, which is made up by the pectate, more specifically calcium and the magnesium pectate. Another one we are talking about primary cell wall. In case of the primary cell wall, obviously these are the thin, as well as whenever we are talking about the fibril, that is cellulose fibril, because हमने just देखा cellulose, these are the fibril part, right? And hemicellulose, that is a matrix part. So cellulose fibril, they present at less concentration, but the part of the matrix, that is hemicellulose, so that hemicellulose they present at high concentration. So as compared to secondary wall, primary cell wall is thin, less concentration of the cellulose fibril, and as well as high concentration of hemicellulose. Okay, and please remember such kind of this condition means primary cell wall is thin. It is very much common. Example, these are the very much common in young plant cell. जितने भी young cells हैं, in that young plant cell, such condition it is very much common. Now cell wall. नाम से पता चल रहा है secondary. It means that they form later. Secondary. So last cell wall pattern is secondary cell wall. As compared to primary cell wall, secondary cell wall is thick. Why thick? Number one, it has high concentration of the cellulose microfibril. Cellulose fibril, first of all, they present in high concentration. Hemicellulose, they present in low. They are present in less concentration. And the very important thing, now inside the secondary wall, there is a deposition of one substance that is lignin. So lignin is present. So cellulose fibril is high, hemicellulose low, and lignin get deposited, and due to that condition, now whatever the cell is become, the cell wall, it is quite thick. So that's why we are talking about the secondary cell wall, because they form at the later stage. जितने भी young cells हैं, young cell में present नहीं होगा, but once that young cell they go towards the maturation, they become adult. So now slowly there is a formation of secondary cell wall. Got it? Clear this point? Any doubt from cell wall composition? Just quickly tell me. So middle lamella, उसके अंदर present होगा primary cell wall, then secondary cell wall, then plasma membrane, and now the cytoplasm, nucleus, organelle part, etc., etc. Any doubt? Manjita, Siddhi, Ramchandra, Sandesh, Vedant, Suhas, Komal, Abhilasha. Chirag, Edric, Jail, Nihal, Sia, okay, chalo. Next part, again neat favorite question, plasmodesmata. Plasmodesmata, also called as cytoplasmic connection. These are also called as the cytoplasmic connection. Normally what happens whenever we are talking about the plasmodesmata, first of all, they are present only and only in case of the plant. First of all, plasmodesmata, it is not present in animal. It's present in plant, number one. Next one, whenever we are talking about the cytoplasmic connection, obviously, they are some minute pore or the small pore inside the cell wall. So when we are talking about the plasmodesmata, these are the very minute pore, small pore inside cell wall. Inside cell wall. Okay, so that connection simply it is called as the plasmodesmata. Now, whenever we are talking about the plasmodesmata, basically these are the also called as the cytoplasmic connection because two cells cytoplasm they get connected with the help of the plasmodesmata. So suppose quickly I am taking the diagrammatic representation how it is exactly look. एक plant cell है and exactly near there is a presence of the another plant cell. Suppose these are the two plant cells. These two plant cells they are getting connected. So you know very well toward the outermost side now the cell wall is present like this. So I'm not talking about all the part. So you can imagine There is a presence of the cell wall like middle lamella, then primary cell wall, then secondary cell wall. So this part, so this part basically it is called as the cell wall. So for the cell wall, I'm writing CW. Outermost layer, which is represented by this white color. Now these are the PM, that is plasma membrane. Now what happened? Cell of the plasma membrane, they are showing connection like this. They are showing the connection, and that is just something a minute pore. That minute pore, it is called as the plasmodesmata. Plasmodesmata. Okay. So if I'm talking about the simple question, plasmodesmata also called as the cytoplasmic connection. It is a simple question as it is. Alternate name cytoplasmic connection because they can connect. Okay, Komal. They can connect two cells cytoplasm with each other. But if I'm talking about on the basis of the origin, so plasmodesmata, how they get originated. So if question that is related to origin, so that is a quite hard question, means hard level question. So that time you have to remember. Normally, what happened? Endoplasmic reticulum is present. Endoplasmic reticulum, that is ER, they showing extension. They showing extension between two cells. They showing extension between two cells. And such kind of this extension, simply these are called as the desmotubules. These are called as desmotubules. Okay. And now what happened? All these desmotubules, whatever they form, finally they form a cytoplasmic connection, and that cytoplasmic connection simply is called as the plasmodesmata. They form plasmodesmata. So if I'm talking about this straightforward question, plasmodesmata also called as the cytoplasmic connection. But is this any question related to origin? So please remember, endoplasmic reticulum which is present in one cell as well as endoplasmic reticulum which is present between the another cell. Now that endoplasmic reticulum, they get elongated, right? They get showing extension. So normally kya hoga dekho, suppose you know very well, endoplasmic reticulum, it is having the some structure, long elongated structure, suppose like this. Suppose these are the endoplasmic reticulum of cell 1, and now these are the another one, endoplasmic reticulum, which is present between the cell 2, suppose. Now randomly, ER of one cell, it is showing connection with ER with the another cell through that plasmodesmata. So you know very well, plasmodesmata, these are the minute pore which is formed between the cell wall, right? And now ये जो structure बना, whatever the structure get formed, the such structure is called as the desmotubules. Such structure is called as the desmotubules. So now what happened? Suppose ये one cell है. Now these are the another cell. Suppose these are the one cell, and now these are the another cell. So ये एक cell हो गया. Now these are the cell second, and whatever this color, ये color है, this one, that is a blue color. Now we are talking about these are the plasmodesmata. So just check the exactly position. Plasmodesmata, now what happened with the help of the plasmodesmata, cytoplasm of cell A, it's directly connected with the cytoplasm of second cell. So whatever we are talking about, this yellow color, these are the plasmodesmata. But now what happened? To maintain the plasmodesmata structure, desmotubules also play a very important role. And what is meaning of the desmotubules? Endoplasmic reticulum, because you know very well, endoplasmic reticulum, it is a plasma membrane. It is made up of the plasma membrane, right? So that plasma membrane of endoplasmic reticulum, they are showing extension to endoplasmic reticulum. Two cells, two endoplasmic reticulum, they join with each other, and with the help of that condition, they can form a typical structure that is called as the desmotubule. So just imagine Desmotubules, it is a plasma membrane extension which is get originated from the endoplasmic reticulum, and that desmotubule, it is simply covered by the plasmodesmata. So with the help of the plasmodesmata, first function, that is desmotubule get formed. Another one, cytoplasmic connection, means one cell cytoplasm, they directly connected with the another cell. So अगर simple question बन रहा है, that time you have to remember plasmodesmata, also called as the cytoplasmic connection, where two cells cytoplasm, they connect with each other. And if I'm talking about the origin, how they get formed, so these are the originated because of the desmotubules. And what is meaning of the desmotubules? Endoplasmic reticulum of the two cells, they are showing extensions, they join with each other, and such kind of this long ER extension, simply it is called as the desmotubules. Okay, so हमने एक simple question पे discuss किया, and we'll discuss one hard question also. Any doubt from this area? Plasmodesmata, cytoplasmic connection, and the desmotubules. Any doubt? Pratyusha. Cytoplasmic connection means they can connect two cells. Cytoplasmic connection, na baba? Okay, chalo. I'm giving the functions. Chalo. So we are talking about function of the plasmodesmata. Number one function, basically they are showing transport from one cell to another cell. Another one, they are also responsible for exchange of chemical between two cells. Between two living cells. Because plasmodesmata, these are the part of the living cell. It is not part of the dead cell. Please remember, the living cell में present होगा. So we are writing a living cell. So simply cytoplasmic connection. Two cells connected with each other, responsible for the transport of material, exchange of the chemical between the two living cells. Then, और हम इसको आगे देखेंगे, third function also, it is also involved during the symplast pathway. It is also involved during the symplast pathway or symplast movement, that is movement with the help of the plasmodesmata. So this part we'll discuss in chapter transport. Done? I hope you remember, if you revise, symplast, apoplast, these are the two pathways where molecules, they can transfer from one cell to another cell. So symplast pathway, nothing but that is movement of molecule with the help of plasmodesmata. Yes, every plasmodesmata, they has the desmotubules. हाँ, yes, Abhilasha. Done. Chalo, any doubt from the plasmodesmata? Any doubt? Okay, other students, any doubt? Yeah, chalo. Next part we are talking about plasma membrane, also called as the cell membrane. Cell wall part is over. Now we are talking about next part, plasma membrane, or also called as the cell membrane. Each and every living cell, obviously, each and every living cell, it is consists of the plasma membrane. Cell wall present होगा या फिर absent होगा. But when we are talking about the plasma membrane, each and every living cell, they has the plasma membrane, because plasma membrane, it is act as a boundary. So when we are talking about the plasma membrane, first of all, these are the very thin, elastic, semi-fluid. Just check your mic. किसी का तो mic on है. Thin, elastic, semi-fluid, as well as these are the protective covering layer. Protective covering layer. Okay. So when we are talking about the protective covering layer, they can cover plasma membrane. These are the part of the organelle also, because cell organelle like mitochondria, chloroplast, endoplasmic reticulum, nucleus, all those organelle they has the plasma membrane, as well as outermost wall of the outermost boundary of the cell, that is cell membrane. So cell membrane also they act as a boundary, because in case of the animal cell, you know very well, cell wall is absent. So plasma membrane, they play very important role. They define the shape, they also involved during the protection. So whenever we are talking about, these are the protective covering layer outside the cell, means outside the cytoplasm, as well as the inside the cytoplasm, means they can cover, they can form boundary to, they can form a layer with all cell organelles, means who has the plasma membrane, who consists of the plasma membrane, as well as they act as the outermost boundary also. Chalo. Next part we are talking about composition. Composition of plasma membrane or composition of the cell membrane. When we are talking about the composition, number one, we are talking about the concentration of the protein. Normally, protein it is present 2 to 70%. When we talk about the lipid concentration is approximately 20 to 79%. It's quite high. Third one, we are talking about sugar part, that is carbohydrate part. So carbohydrate part is quite less. It has only 1 to 5%. Along with that, we are talking about there is a presence of the enzymes. Enzymes also they get associated. And I'm talking about approximately 30 enzymes, 30. Approximately 30 enzymes, they are associated with plasma membrane. Associated with the plasma membrane. And now we are talking about last component, that is water. Water also, these are the one component of the plasma membrane. So approximately 30 enzymes are there, and water also present, and approximately of water concentration it's like approximately 20%. So हम major focus करेंगे with three parts: protein, lipid and the carbohydrate, because these are the three component, major component which is a part of the cell membrane. So first of all, we start with the first part, that is protein part. Protein. चलो. No need to remember percentage, because वहाँ से question नहीं बनेगा. Don't worry. But हाँ, there will be one question on the fluid mosaic model. So to understand the fluid mosaic model, we have to understand protein, protein types, lipid, as well as the carbohydrate. No crystallines moving there. It's fine. Yeah. Just check your internet connectivity. Okay, chalo. Should I start with protein? So we are talking about protein. Basically, there are two types, depend upon their extraction. So one protein that is external protein, another protein that is internal protein. We are talking about the types of protein. That is number one, that is internal protein. एक second. Don't worry if you miss any line or any sentence. Once class will be finished, I'll share the whiteboard link, entire link. I convert into PDF and I'll share you. So you can complete your notebook also. Okay? Yeah. Thank you. So next part, we are talking about part of the protein. Internal protein also called as the intrinsic protein. and intrinsic protein also called as the integral protein. Integral protein नाम से पता चल रहा है they reside inside. And another part we are talking about protein, that is peripheral protein, which is present on the periphery, which is present on the surface. Such type of this protein also called as the extrinsic protein, and extrinsic protein also called as the external protein. So you know very well, out of, if you want to isolate protein from the plasma membrane, isolation with the help of any chemical or by just change or by applying any alkali or the acidic solution or just alter the pH. So the protein who attach on the periphery, the protein who attach on the external side, such type of the protein, they are very easy to remove. It's very easy to remove because now what happened, they attach on the periphery. So on the basis of that, definitely as compared to intrinsic protein, peripheral protein, they are very easy to remove. Okay. Next part we are talking about types of protein on the basis of their function, which is based on the function. So you know very well, whenever we are talking about the protein part, obviously that protein number one, they can act as a structural protein. First of all, first function, they act as a structural protein. And you know very well, whenever we are using the word structure, it means that they can form a backbone. They can act as a support. They can form a backbone. So such kind of this protein, they can form a structural protein. Sometimes protein, they act as a carrier protein. Carrier from one place to another place. And I hope you remember, whenever we are using the carrier protein, so whatever we are talking about, the transmembrane protein or something, tunnel proteins are there. So they are just responsible for movement of molecule from one side to another side, because plasma membrane is act as a barrier. It's the selectively permeable. So now whenever we are talking about the carrier protein, major function with respect to carrier protein, they are responsible for the transport. So we'll discuss later form what kind of the protein and how they're responsible for the transportation. So major function with respect to carrier protein, they're responsible for the transport. And in some parameter, they are also act as a some enzymatic activity, because you know very well, all enzymes, they are protein in nature. All enzymes. Whenever we are talking about the another function, that is another type of the protein, that is enzymatic protein. And whenever we are talking about the enzymatic protein, obviously they are catalytic in nature, means they can responsible for the various reaction, catabolism or anabolism. So catalytic in nature, catalytic means what, they involve during the reactions. So again, these are the three types of the protein. Major protein, which is based upon their function, structural protein. Another one, that is carrier protein, and the next one, that is enzymatic protein. Done. Any doubt from the protein part? Very simple, because we'll discuss quickly fluid mosaic model. So in fluid mosaic model, we'll try to mention all these things. Chalo. After the protein part, now we are talking about the second component, that is lipid. Second component, that is lipid. I hope you remember, we already discussed this part in Kingdom Monera. In case of this, whenever we are talking about the lipid, it has the head portion as well as it has the tail region, head and the tail. So this part basically is called as the head of the lipid, and the lower part, these are called as the tail of the lipid. Head, basically they are hydrophilic in nature, means they can interact with water, so hydrophilic. And when we are talking about the tail nothing but these are the fatty acid chain, long chain of fatty acid. That's why we are using the tail word. So tail, basically they repel from the water, and that's why we can say these are the hydrophobic in nature. We'll discuss that part later. Whenever we are talking about the typical arrangement of the plasma membrane, head always present toward the outer side. They always face to water, while tail, they are embedded inside. They present at the core region. They repel from the water. But when we are talking about the part of the lipid, so in case of the lipid, you know very well, might be possible. Few examples. There is a presence of the phospholipid also. That is lipid who attach, who conjugated with the phosphate group, phospholipid. And you can take some example of the phospholipid like cephalin. Cephalin is example of the phospholipid as well as the lecithin. Lecithin also, these are the one example of the phospholipid where lipid present definitely, but along with that lipid, phosphate group is attached. Some carbon, hydrogen, oxygen number get vary, and now they can form a phospholipid. The two example very simple, cephalin and the lecithin. The rest portion you will study in biomolecule chapter. Now, second component which is again come under the lipid category, that is we are talking about cholesterol. You know very well, cholesterol actually इसके बहुत सारे levels होते हैं. There is a bad cholesterol also, there is a good cholesterol also. So means LDL, then low density lipoprotein, high density lipoprotein, very high density lipoprotein. But cholesterol, it is good thing if it is present in optimum concentration, because cholesterol, it is very important, because cholesterol, they provide stability to plasma membrane. Stability is act as a stabilizer. Even in case of the bacteria also, not cholesterol, but cholesterol like substance also present in case of the bacterial plasma membrane. Even in case of the plant plasma membrane also, cholesterol like substance are present. But remember this point, cholesterol, these are the present only and only in case of the animal plasma membrane. ना ही वो plant में होता है, ना ही वो bacteria में होता है. But हाँ, some other molecule which is functionally similar to cholesterol, those are present in bacterial plasma membrane as well as present in plant plasma membrane. So here we are talking about the molecule cholesterol, 30 carbon, and such cholesterol, they provide stability. Okay, which is again one form of the lipid. And the next one, sometimes lipid, they are showing conjugated with carbohydrate. So for example, I'm taking some example, that is glycolipid or the galactolipid. So for example, galactolipid, and you know very well, galactolipid means what? Sugar, that is galactose, which is a carbohydrate, right? And now such galactose, they get conjugated with the lipid molecule. So lipid, they can bind with the protein, lipid, they can bind with the carbohydrate. So simply we are talking about carbohydrate plus lipid composition. And simple example, that is galactolipid, because lipid, they has tendency, they can bind with protein, they can bind with carbohydrate, or might be possible they can bind with phosphate group also. Okay, means they can become more complex, require stability. That time they become more complex. So these are the very simple information with respect to lipid part. Okay, and the last part we are talking about the part of the carbohydrate. Which very less concentration, 1 to 5%. Now when we are talking about the carbohydrate, बहुत सारे carbohydrate होते हैं, like for example simple sugar present है, like hexose sugar without any combination, hexose is present. Sometimes hexose, they get conjugated with the amine group, that is hexosamine. Sometimes, not always. Okay, so hexosamine means 6 carbon sugar, they get conjugated with the amine group, conjugated with the NH2 group. So we can say that is sugar plus amine. यहाँ पे present है only sugar. And sometimes they can form some modified sugar also, like sialic acid. Sialic acid, and sialic acid means what? Something, it is an acidic sugar. These are one example for the acidic sugar. So it depended. So whenever we are talking about the part of the carbohydrate, simple carbohydrate also present. Sometimes carbohydrate, they get conjugated with phosphate group also, they get conjugated with amine group also, sometimes they become acidic also. So different types of the carbohydrate are present. But please remember, in case of the carbohydrate part, when will we start with oligosaccharide? Because you know very well, carbohydrate के बहुत सारे types थे, like monosaccharide, disaccharide, oligosaccharide, polysaccharide, homopolysaccharide, heteropolysaccharide, बहुत सारे types है. But when specifically we start with the word oligosaccharide, okay, oligosaccharide. So please remember, when we are talking about the oligosaccharide, simple meaning, that is sugar which is associated with sugar which is associated with lipid as well as the protein. Dependent, they can bind with the protein or might be possible they can bind with the lipid. अगर ऐसे हुआ, suppose that oligosaccharide, that is sugar part, which is react or which is bind with the lipid. Suppose sugar plus lipid, carbohydrate plus lipid. So that time such kind of the structure simply it is called as the glycolipid. Such kind of the structure simply it is called as the glycolipid. And sometimes carbohydrate, that is sugar part, also they get joined with the protein, and such kind of the structure simply they can form a glycoprotein. So it's very much common during the oligosaccharide part, formation of the glycolipid and as well as the formation of the glycoprotein, they are very much common. But please remember one important question: when you talk about glycolipid, when you talk about the glycoprotein, or when you talk about the oligosaccharide, they are always present toward the outer side of the membrane. Outer side, okay, always they present toward the outer side, because sometimes what happen, when we talk about the function of the oligosaccharide, now uska major function hota hai, they has the antigenic property. Antigenic property means what? For example, we are talking about lambda phage virus. लैम्बडा फाज दे स्पेसिफिकली इन्फेक्ट टू ई कोलाई बैक्टीरिया. ना ही वो बैसिलस को इन्फेक्ट करता है, ना ही वो ईस्ट को इन्फेक्ट करता है, ना ही वो हमें इन्फेक्ट करता है. व्हाई? बिकॉज ई कोलाई दैट इज अ होस्ट. सो हाउ lambda phage recognize is this E. coli bacteria or is this bacillus? So whatever they toward the outer side of the E. coli body, some sugars are present. Sugars they get conjugated with protein, or sugar they get conjugated with the lipid, like glycolipid, glycoprotein, or commonly you can say that is a oligosaccharide. So before infection, viruses or any phage, they can recognize the oligosaccharide, because oligosaccharide is the identity of that organism. Corona virus भी वैसे ही होगा. Why he specifically infect to mammals in the sense human? Because during the infection, means during the spreading process, during their life cycle, they require the host body. So before what we said, before the entry, now he simply recognize what kind of the protein, what kind of the oligosaccharide, glycoprotein or the glycolipid, those are present toward the outer side. तो पहले वो रिकॉग्नाइज करेगा, पहले चेक करेगा ये मेरा ही होस्ट है ना, मैं अंदर जाऊं या ना जाऊं. So after that they start multiplication, because you know that virus, these are the host specific, right? And before the entry, virus always check ये मेरा होस्ट है या फिर नहीं है. So oligosaccharide, very important step when we are talking about the oligosaccharide or the glycoprotein or the glycolipid. Please remember they are present only present towards outer membrane. Always. Question. Okay, they are always present toward the outer membrane, because when we are talking about the oligosaccharide, they can bind with what we say, they can attach with the lipid as well as the protein. So we are talking about such kind of this. So we are talking about next condition. So when we are talking about such kind of this glycolipid plus such kind of this glycoprotein as well as the glycolipid, further they can form a glycocalyx. They form a glycocalyx. Okay, and if I'm talking about what is the exactly function of the glycocalyx, so glycocalyx, first of all, it is help in attachment, because sticky substance, glycocalyx, right? So it is help in the attachment, as well as just we discuss, it show antigenic property. And the next one, These are the also a recognition center, self and the non-self. It is also act as a recognition center. So please remember one point with respect to oligosaccharide. These are the sugar associated with the lipid and the protein. Glycolipid and the glycoprotein collectively they form a layer of the glycocalyx. हमने एक kingdom में देखा था, kingdom under the kingdom monera. Glycocalyx functions, they help in the attachment as well as they are showing the antigenic property and as well as they can act as a recognition center. Got it? Clear? Okay, chalo. So quickly within 2 minutes, we are taking the last point. Model for the plasma membrane. बहुत सारे models है actually, but most widely accepted model for the plasma membrane, that is fluid mosaic model. Fluid mosaic model, and such fluid mosaic model it is given by, it is introduced by the two scientists, scientist Singer and the Nicholson. Question. And Nicholson, which is a widely accepted model for plasma membrane. Okay, fluid mosaic model. So quickly we try to mention oligosaccharide, even intrinsic protein, extrinsic protein, lipid, cholesterol, everything. Chalo. So we are trying to draw such structure. So lipid, it has the part of the head, tail. So head and tail, these are the simple structure for the lipid. Now we are talking about one protein who occupy the two layer, and such name of the protein that is called as the tunnel protein. Tunnel. तो ये पहला प्रोटीन हम देख रहे हैं, first one. We are talking about name of the protein, that is tunnel protein. Why we are talking using the word tunnel? Because you know very well, tunnel they can connected the two. For example, they can connected with the two region. So definitely when we are talking about such kind of this tunnel protein, they can cross two layers, because plasma membrane is a bilayer, outer membrane, outer layer and the inner layer. So they can cross the two layers. So tunnel protein, and tunnel protein also called as the transmembrane protein. Also called as the transmembrane protein. Okay. Now we are talking about, there is a presence of the cholesterol. Cholesterol basically they are present, they provide the stability. So they are associated with the lipid structure. So whatever we draw here with the help of this blue color, that is called as the molecule, that is cholesterol. And you know very well, cholesterol basically is a membrane stabilizer. They provide the stability. So it's the membrane stabilizer. Next one, we are talking about protein, that is extrinsic and the intrinsic protein. So just we discuss extrinsic protein, which is present towards the periphery, like this. Both the side they are present. So I'm just writing E, and E stand for the protein, that is extrinsic protein, which is present toward the outer side, peripheral protein, which is easy to remove. Next one we are talking about, that is intrinsic protein, and concentration of the intrinsic protein, it is always more as compared to extrinsic. So if you want the concentration of the extrinsic protein, that is 30%, and the intrinsic protein concentration is quite more, approximately 70%. So suppose we'll try to draw intrinsic protein. This red color. Get lucky. Just 1 second. Hmm. So extrinsic protein. Now we are talking about next protein, that is intrinsic protein. Huh, Komal? It's now opening. Don't worry. So this red color is example for the I. I, that is intrinsic protein. So I'm just writing I, that is I. I stand for the protein, that is intrinsic protein. And intrinsic protein concentration is approximately quite high, that is 70%. Okay, so we'll draw tunnel protein, intrinsic protein, cholesterol, as well as the extrinsic protein. Final point, we are talking about presence of oligosaccharide. Okay, so remember this point, oligosaccharide, they are always present toward the outermost side. So that oligosaccharide simply represented by like this, which is attached with the extrinsic protein also. Oligosaccharide, they can attach with the lipid molecule also, because they can form a glycolipid then. And sometimes that intrinsic oligosaccharide, they are showing attachment to intrinsic protein also. So please make sure these are the structure, these are called as the oligosaccharide, and oligosaccharide, they are showing attachment with the external protein, attached with the internal protein, as well as they are attached with the lipid also. So that's why we'll discuss one point, they can form glycolipid as well as the glycoprotein. And if I'm talking about exactly position, so glyco oligosaccharide, they are always and always present towards outer membrane. Always, please remember, not toward the inner membrane. It is always and always they present toward the outer membrane. Okay, and whatever we are talking about this side and this side, so plasma membrane basically it is a bilayer. So ये बना हमारा outer layer, and this one the inner layer, because plasma membrane is a bilayer structure. Okay, yeah. So these are the exactly very simple, very basic, most accepted model, fluid mosaic model, where we draw tunnel protein also, we'll mention extrinsic protein also, intrinsic protein also, cholesterol, and as well as the most important thing, one question, oligosaccharide, which is always and always present toward the outer side of the membrane. Okay, done. Abhilasha, Atul, Chirag, Chrisley, Deepika, Dipasha, Dipasha, yeah, Hedrick, Gargi, Gautami, Hayden, Ishika, Jared, Jail, Komal, मंजिता, नैनी, नेहा, पूजा, प्रत्युषा, प्रियांशु, राखी, रामचंद्र, रिजन, रेम्बेली, रिया, संदेश, शिवांगी, श्वेताली, सिद्धि, सिया, दो सियाएं, सुनिधि, सूरज, स्वप्नाली, स्विजेल, तन्वी, तुषा, ओके, वेदांत, वेरॉन, विनिया. It's Yumi, if I'm not pronounced wrong. Okay, yeah. So from next class, we'll start with the next organelle, mitochondria and chloroplast. And again, I think one question is already asked in 2016. उसके बाद question बना था वहाँ से 2018. उसके बाद नहीं question बना. So mitochondrial chloroplast part also important because two important concepts associated with. Number one is the semi-autonomous organelle, and the number second they are showing endosymbiotic association. So this part we'll discuss in next class. Okay.